Hey everyone, Dr. D here, and in this video, we are covering Chapter 23 from our Talaro's Foundations in Microbiology 12th edition textbook. So let's go ahead and get started.
Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. Explain stuff.
All right, Chapter 23, we're talking about parasites. And the parasites we're referring to are the worms, remember the helminths, as well as the protozoa. That's what we mean by parasites in this chapter. And we're going to talk about those parasites of medical importance. In fact, if any of you have ever thought of taking a parasitology course in college, that's what you'll be focusing on are these eukaryotic parasites. We're talking, again, protozoa, helminths, the cause of 20% of infectious diseases. These are, of course, now much less prevalent in industrialized first world countries, but they are still quite common in developing areas. So let's start with the protozoan pathogens. Remember that the protozoa are single cell animal like microbes. Do you know what they mean by animal like? They mean that they ingest their food. Like you and I eat, we're heterotrophs, so we're animal like. These eukaryotic single cell organisms, they consume organic molecules to grow. Like you and I consume sugars and fats, they also consume sugars and fats to grow. They are not photosynthetic. That would be plant like, right? So think of it this way. Think of it this way. Do you guys remember the protists? We talked about protists. There are two branches of protists. And remember, the animal like ones are called the protozoans or protozoa, and the plant-like ones are the algae. Think of it that way. So that's what we mean by animal-like microbes, most having some form of motility. Do you guys remember there are amotile protozoans, flagellated protozoans, ciliated, and amoeboid with pseudopods, right? Those are the four classes based on their motility. You should make a note that protozoa are classified based on their motility. Are you a flagellate? Are you a ciliate? Are you amotile? Do you move around with pseudopods? Well, that's your motility class. There's an estimated 100,000 species, but approximately 25 are important pathogens. Remember, pathogens cause disease. They have a variety of life cycles. Most of them propagate by simple asexual cell division. They have a motile active feeding stage called the trophozoite stage, but many can also become a cyst and a dormant cyst stage. Others can have complex life cycles that include both asexual and sexual phases. On this table, I just want you to take a glance here and notice that different protozoans are classified, again, based on their motility. So take a look here. There are amoeboid protozoa that move around with pseudopods. Remember those fake feet extensions of the plasma membrane? And you see here Entamoeba histolytica, Naegleria, Acath amoeba. These are all examples of amoeboid protozoa. Next, look at the ciliated protozoa. Balatidium coli is a ciliated protozoa. And then look at flagellated protozoa. Giardia, Trichomonas, Trypanosoma, Leishmania. And then there are the apicomplexan protozoans. These are ones that don't have a specific type of motility usually, like Plasmodium, which causes malaria. So let's go into a little bit more detail about Entamoeba histolytica and Amebiasis. This species of amoeba alternates between the motile trophozoite stage and the smaller non-motile cyst dormant stage. And oh, here's Punky. Punky's come to say hi, everyone. Look how big she's gotten over just the last couple of weeks. She's large and in charge now. Hi, Punky. She's purring. All right, she's going to probably join me for the rest of this section. I don't know if you can hear her purr. Maybe. All right. Now, the trophozoite has a large nucleus and lacks most other organelles, while that mature cyst that it forms is encased in a thin, tough wall and contains 4 nuclei and bodies called chromatoidals, dense clusters of ribosomes. And here's the life cycle of this Entamoeba histolytica and how it causes the disease, Amebiasis. First, the amoeba is secreted in the feces, and you can have either the trophozoites secreted or the cysts secreted in the feces. And by the way, that's how you diagnose someone with the disease. You take a stool sample and you look for the presence of these trophozoites or cysts. And then infection occurs when you ingest one of these cysts. You ingest from fecally contaminated food, water, or hands. And it's those cysts that give you the disease. The trophozoites that are secreted in the feces, they don't last long outside of the body. And even if you did ingest them, those stomach acids would destroy them. So it's really the mature cysts that spread the disease, not the shed trophozoites. Once the cyst makes it past the stomach, there's a process known as excistation, which means the cyst grows back out into the trophozoite state. Next, those trophozoites multiply through binary fission, and that produces both trophozoites and cysts. And again, those cysts and those trophozoites, they pass again in the feces. And the cysts that leave the body in the feces can survive for days to weeks outside the body and remain infectious in the environment due to that protective cell wall. The intestinal disease occurs when trophozoites invade the intestinal mucosa, and also extra intestinal disease can occur if the trophozoites leave the intestines through the bloodstream and end up at different parts of the body, like the liver, the lungs, and even the brain. And yes, us humans are the primary hosts for amoebiasis. Those trophozoites spread in our body or in our intestines. They attach, multiply, and actively move about and feed. 90% of patients with this particular amoeba are asymptomatic. However, the amoeba may secrete enzymes that dissolve tissues and penetrate deeper layers of the mucosa, causing dysentery. Remember, that's bloody diarrhea, abdominal pain, fever, diarrhea, weight loss, and it's carried by about 10% of that world's population. Here you can see the appearance of amoebiasis and dysentery lesions. These are dysentery lesions or erosions of the intestine caused by damage to the intestinal mucosa by this particular amoeba. It can even become life-threatening if it manifests as hemorrhaging, which is bleeding, perforation, which is holes, appendicitis, which is inflammation in the appendix, and tumor-like growths. Remember I mentioned that these amoeba might invade the liver, the lung, and the severe forms can even have a 10% fatality rate. Remember, how do you diagnose for this disease? You take a stool sample or a fecal smear. The presence of either trophozoites or cysts in this smear are indicative of having that amoeba. Treatment include these anti-protozoal medications listed here. Here you can see a picture of Entamoeba histolytica. Here it is in its trophozoite stage, filled with its primary nutrient, red blood cells. It phagocytoses red blood cells and feeds on them. You could see how this would result in that fever and all of those other symptoms and signs of the disease. And on the right, you can see the cyst stage of Entamoeba histolytica, and you can see it's got the 3 nuclei. You see the 3 nuclei with the black arrows, and the chromatoidal, which is the red arrow. Remember, that's a cluster of ribosomes. Next, let's talk about amoeba and the brain. There are a couple of amoebas that are known for getting into the human brain and causing death. You may have heard of brain-eating amoeba. Have you ever heard of that? We actually have that here in Texas where I live, in warm water, like the lakes, the local lakes. They possess this amoeba. And the way you get this amoeba disease is by jumping in, and you acquire the amoeba through the nasal canal. Basically, you if you splash into the lake and water splashes up your nose, if those amoeba were in that water, they would make their way up into the nose. And those um, inside of the nose are your sinuses, and there are certain neurons that go from the sinuses directly to the brain. And these amoeba, they just love to travel along those sinal canals, along those neurons, straight to the brain, where they cause massive brain damage. And infiltration of the brain is usually fatal. So that's why they call it the brain-eating disease. And these are caused by Naegleria fowleri and Entamoeba. On the left, you can see the creature Naegleria fowleri with its thick, blunt pseudopods, and it is filled with food vacuoles in this image. On the right, you can see Entamoeba, which has spine-like processes extended from its pseudopodia. Either one can make its way to the human brain and cause that encephalitis. It's called meningoencephalitis, which is massive swelling, primary acute meningoencephalitis, massive swelling and inflammation and infection in the brain, and that's what leads to death. So this is why they say if you go to the warmer lakes in some of those warmer regions of the US, you should always plug your nose before you do a cannonball into the water, especially if you're in the murky, shallow end of the lake where there's like muddy, because these amoeba live in the mud. And if the mud's kicked up and that muddy water makes it up into your nose, that's a sure way of getting this brain-eating amoeba. Next, an intestinal ciliate. Now we're moving on from the amoeba. Now we're talking about ciliated protozoa like B. coli or Balatidium coli. It's an occupant of the intestines of domestic animals such as pigs and cattle. And how do you get B. coli? You acquire it by ingesting cyst-containing food or water, from their fecal contamination. The trophozoite erodes your intestines and elicits intestinal symptoms, but rarely penetrates the intestine. So unlike Entamoeba histolytica, it's not known for leaving the intestines and going extra-intestinal to the lungs or the brain or something like that. This one stays put in the intestines and causes its disease there. Here you can see a couple drawings depicting its trophozoite stage on the left and its dormant mature cyst stage on the right. Notice the tufts of cilia on the trophozoite stage. Again, this is a ciliated organism. And these parasites, they make their way to the intestines. And did you know your intestines are anaerobic, which means that they are devoid of oxygen? And they live in your intestines among your own bacterial microbiota. They live in and they live amongst your own microbiota. Healthy humans are resistant to infection. However, if you're compromised, then these B. coli can cause intestinal disease. Treatments include these antimicrobials listed here. And that leads us to concept check number 1. Yay! Entamoeba histolytica primarily invades what? Remember, it was the small intestine. It was the small intestine. Though it could go extra intestinal. Remember, it could leave the intestine as well. But it's known for invading the mucosa of that small intestine. Next, let's talk about the flagellated protozoa. We're talking about Trichomonas. These are small, pear-shaped protozoa with 4 anterior flagella. That means 4 flagella in the front and an undulating membrane. These do not form cysts. They exist only in that trophozoite form. There are 3 of them that infect humans: Trichomonas vaginalis, Trichomonas tenax, and Trichomonas hominis. Here we can see a nice image of Trichomonas vaginalis. This is a urogenital pathogen. Here you can see the 4 flagella, the anterior flagella, as well as that undulating membrane here. There's a nucleus and the axostyle. And again, remember, this Trichomonas vaginalis infects the urogenital tract. It causes an STD, or sexually transmitted disease, called Trichomoniasis. The reservoir, remember, reservoir means where you normally find this creature. The reservoir is the human urogenital tract. 70% of infected people are asymptomatic. It is a strict parasite, which means this creature cannot survive for long outside of the host. There are 3 million cases yearly, so it's one of the top STDs. In females, it produces a foul-smelling green to yellow discharge, vulvitis, cervicitis, urinary frequency, and pain. In males, the symptoms include urethritis, thin, milky discharge, and occasionally prostate infection. Treatment includes this anti-protozoal here. Moving on to another flagellated protozoa. This is called Giardia. You may have heard of Giardia. Giardia intestinalis. Also, there's Giardia lamblia. These cause giardiasis. This little creature looks so cute. It has a unique symmetrical heart shape with concave ventral surface that acts like a suction cup. And actually looks like it has 2 little eyes as well, because it has 2 nuclei. So it looks like it has 2 little eyeballs. It's really cute looking cell. I'll show it to you in a second. It can form cysts. The cysts are small and compact and multinucleate. That means they have multiple nuclei. The reservoirs include beavers, cattle, coyotes, cats, and humans. The cysts can survive for 2 months in the environment. Here you can see what I was talking about, these cute little cells. They look like they have 2 little eyeballs, but those are the nuclei. There's 2 nuclei. And this is what the trophozoite looks like. It's a heart-shaped cell. I kind of think it looks like a horseshoe crab. What do you guys think? Doesn't it look like a horseshoe crab? And here's a picture of this little guy on the right. You can't really see its little nuclei very well in this picture on the right, but here's what the cyst looks like in the middle. See, it's a multinucleated cyst. And how do you get giardiasis? How do you pick up this giardia? Usually it's ingested with water or food. The infectious dose is 10 to 100 cysts. That's the number of cysts you have to ingest to get the infection. The cysts enter the duodenum, germinate, and then to the jejunum to feed and multiply. This is where they cause giardiasis, which results in diarrhea, abdominal pain. By the way, they call this traveler's diarrhea too. When you go to another country and they say, don't drink the water, they're talking about picking up this Giardia. And this is a very common cause of traveler's diarrhea. Diagnosis is difficult because the organism is shed in the feces intermittently, not constantly. Treatment includes these antiprotozoals. The agent is killed by boiling ozone and iodine, so it's best to treat your water before you drink if it's not already pre-treated water. Here's an example of using fluorescence in situ hybridization or FISH technology in order to identify Giardia properly. Next, we're still talking about flagellated protozoa, but now we're talking about the hemoflagellates, vector-borne blood parasites. These are obligate parasites that live in the blood and tissues of hosts, human hosts. They cause life-threatening and debilitating zoonoses. Can you beat Wicket? Do you remember what a zoonosis is? That's right, Wicket. I should have asked Punky. She's right here. But that's right, Wicket. A zoonosis is a disease that's spread from an animal. So if you pick up a disease from an animal source, if the reservoir is an animal and it spreads to humans, it's zoonoses. They spread in specific tropical regions by blood-sucking insects that serve as intermediate hosts. These typically have a complicated life cycle where they undergo morphological changes. That means one part of its life cycle might look completely different than another part of its life cycle. And those life cycles are categorized according to their cellular and infectious stages. Members of this group include the Trypanosoma and Leishmania. And here are some of those stages I was talking about. These hemoflagellates have stages. The amastigote is the form lacking a free flagellum. The promastigote is the stage bearing a single anterior flagellum. Epimastigote, this is the flagellate stage. And trypomastigote, the large, fully formed stage. Here you can see exactly what those stages might look like. So let's start with the Trypanosoma species. Trypanosoma are distinguished by their infective stage. The trypomastigote is an elongate, spindle-shaped cell with tapered ends. It's eel-like motility. There are two of these that we're going to talk about, two of these trypanosomal diseases. Let's start with Trypanosoma brucei. This is the cause of sleeping sickness or African sleeping sickness. It's spread by the tsetse fly, which is a little fly-looking creature. There are two variants of the disease caused by two subspecies. There is subspecies Trypanosoma gambiense, which is the Gambian strain of West Africa, and Trypanosoma rhodesiense, which is the Rhodesian strain of East Africa. When the tsetse fly bites, it inoculates your skin with those trypomastigotes, which multiply in your blood and damage your spleen, lymph nodes, and brain. This is a blood pathogen. So you find these little guys inside of your bloods, inside of your blood. On the right, take a look. You can see these little pink circles are your red blood cells. And it's those Trypanosoma brucei that live among your cells. They're not intracellular parasites. They don't live inside of your red blood cells. They live among your red blood cells. They live in there. It's a blood-borne pathogen. And here you can see how Gambiense, the subspecies Gambiense, is at West Africa, while Rhodesiense exists at East Africa. And again, remember the tsetse fly, it bites you. The saliva of that tsetse fly is infected with Trypanosoma brucei. It inoculates the human bloodstream. The parasite matures and invades various organs. In time, its cumulative effects cause central nervous system damage. The trypanosome is then spread to other hosts through another fly, in whose alimentary tract the parasite completes a series of developmental stages. So that means that this Trypanosoma brucei is harbored by mammals. Chronic diseases include sleep disturbances, tremors, paralysis, coma even. How do you diagnose this? The trypanosomes are readily demonstrated in blood, spinal fluid, or lymph nodes. So you would do a blood smear, and then you would look among the little red blood cells, and you would see the trypanosoma. See, here you can see it's this long whip-like structure, right? The trypomastigote. This is that trypomastigote structure, and you see how it lives among the red blood cells in a blood sample. This is what that tsetse fly looks like, and it's a biting fly. And treatment involves these anti-protozoal agents. Control involves eliminating tsetse fly. Next, let's talk about another species of Trypanosoma. It's called Trypanosoma cruzi, and this one causes Chagas disease. It's not spread by tsetse fly. It's spread by the reduvilid bug, or kissing beetle it's called, which is the vector. This particular disease is localized to Central and South America, Mexico, and even southern parts of the US. This kissing beetle is the vector. It possesses the trypanosoma. When the bug bites, it defecates, so the feces contain the Trypanosoma cruzi. So the way this works is this little kissing beetle, it's called the kissing bug because it bites you around your mouth area. And when it bites, it also usually defecates, so it poops. And if you scratch that itch and you introduce that poop into that wound that's left over with the kiss, the bug bite, that can introduce this trypanosoma into your bloodstream. This could lead to a local lesion, fever, and swelling of lymph nodes, spleen, and liver. Here is the life cycle of Trypanosoma cruzi, or Chagas disease, which it causes. Here's a simplified view of that transmission cycle. Here is that reduvilid bug, the kissing beetle. Its scientific name is Triatoma infestans. This is the little kissing beetle. And remember, that kissing beetle contains the Trypanosoma cruzi flagellate protozoa. It then bites you around the mouth area and it defecates. And remember, that's how you pick up the flagellate. That's how it gets into your blood. And it's that trypomastigote stage that's found inside of the beetle. This is passed to humans or other mammals. When the bug takes a blood meal, the trypomastigotes then transform into amastigotes and multiply within infected tissues. That's what causes the symptoms of Chagas disease. It's when that reduvilid bug bites an infected human, those amastigotes pass from the human to the bug, completing that life cycle. And unfortunately, with Trypanosoma cruzi, it can kind of go dormant for years or even sometimes decades. Heart muscle and large intestines harbor masses of these amastigotes. It can lead to chronic inflammation, especially in the heart and brains. It can even lead to heart attack and death if not diagnosed and if left for years. These are the anti-protozoals that are used to treat the infection. But again, the problem is usually when you get bit, you don't realize you were bit. You might have a fever and then it might clear. And then these amastigotes, they basically live dormant inside of your tissues. And And then these amastigotes get into the spaces between your muscle fibers, and they get into different tissues, and they can cause disease over decades, and that could culminate in a heart attack or something serious later down the road. So on the left, you can see a sign of acute infection is Romane's sign, swelling of the eye or mouth at the point of the pathogen. See, right where the pathogen bit around the mouth, there's swelling. On the right, you can see a section of heart muscle with those amastigotes. Let's see, these little amastigotes are filling up these spaces between the muscle fibers and the heart. You can imagine how dangerous this is to have this little creature growing inside of your heart. Is going to culminate in uh, cardiac damage. Next, let's talk about another flagellate, Leishmania species, which causes Leishmaniasis. Leishmaniasis is a zoonosis. Remember, that's transmitted from mammalian hosts, animals to people. And the vector is the female sandfly. Remember, vectors are living transmitters of disease. This sandfly requires a blood meal to produce eggs. It's endemic to equatorial regions. So in this case, the promastigotes are injected by that sandfly bite, and then they convert to amastigotes and multiply. See, the sandfly becomes infected by biting a host. The promastigote stages in the gut of the sandfly. The sandfly bites the host, either a human host or a mammalian host. Inside of the host, it becomes the amastigote inside of a macrophage, and they live inside of macrophages. So you've got these amastigotes living within macrophages and outside of macrophages. If the macrophage does not migrate, the infection is localized. If the macrophage does migrate, it becomes systemic. So Leishmaniasis can culminate in different ways. It could form a cutaneous localized sore. It could culminate in espundia, skin and mucous membranes infected of the head, chronic infection. It can go systemic or visceral with high intermittent fever, weight loss, enlarged spleen, liver, and lymph nodes. And Kala-Azar is the most severe and fatal form of this systemic infection if untreated. Here you can see cutaneous leishmaniasis. See how it's an ulcer, a localized skin ulcer on the ankle. All right, concept check number 3. Hemoflagellates are transmitted by insect vectors. Remember, like the tsetse fly or the sandfly or the kissing beetle. All right, and lastly, remember the last motility class for the protozoa? We're talking about the apicomplexans, the ones that are considered amodal. These are sporozoans. They lack locomotor organelles in the trophozoite state, so they are known as the amodal protozoa. They alternate between sexual and asexual phases and between different animal hosts. Most form specialized infective bodies that are transmitted by arthropod vectors, these biting insects. They can also be transmitted by food, water, or other means. These include Plasmodium, Toxoplasma, and Cryptosporidium. Let's start with Plasmodium. This is the agent of malaria. You've heard of malaria, right? The disease. It's caused by Plasmodium. It's an obligate intracellular sporozoan. There are 5 species: Plasmodium malariae, Plasmodium vivax, Plasmodium falciparum, Plasmodium ovale, and Plasmodium knowlesi. It's the female Anopheles mosquito that is the primary vector. You can also pick up these Plasmodium via blood transfusions and even mother to fetus. This is responsible for 200 million cases each year, with almost 500,000 deaths per year. And this is along what's called the malaria belt. You see the pink zones along the equator? The pink zones outline the major regions that harbor malaria. The malaria life cycle is interesting. There's an asexual phase within the human host. Infected female Anopheles mosquitoes inject asexual sporozoites, which then travel to our liver. Then through schizogony, asexual division, you generate merozoites. The merozoites enter circulation in 5 to 16 days, depending on the species. Those merozoites attach to and enter the red blood cells. They literally get inside of your red blood cells, where they convert into trophozoites and multiply. The red blood cell at some point bursts open, releasing all these merozoites that differentiate into gametes. This leads to the sexual phase, which is inside of the mosquito vector. The mosquito draws infected red blood cells during its blood meal. Gametes then fertilize, forming a diploid cell. They make a zygote, or more specifically, a sporozoite in the stomach. Those sporozoites lodge in the salivary gland of that mosquito, and that's available to infect the next human host. So isn't that neat? When the mosquito draws the red blood cells, those sporozoites form. The sporozoites lodge into the salivary glands, the saliva glands of the mosquito. So the next time the mosquito bites a human and it, spits up some of that Novocaine, that dulling solution it has, so you don't feel pain. That's how it infects you, through its salivary glands. That's how the protozoa gets into your bloodstream. Now, once you have malaria, symptoms include episodes of chills, fever, sweating, anemia, and organ enlargement. Symptoms occur at 48 to 72-hour intervals as red blood cells keep rupturing. Literally, I've seen this under a microscope. Your red blood cells pop open and all these merozoites burst out of your red blood cells, and that cannot be good for your immune system. This is what leads to all that fever and sweating and chills and anemia. Your red blood cells are literally popping. And it's Plasmodium falciparum, which is the most malignant type with the highest death rate, especially in children. And how do you diagnose malaria? Well, you have to find the presence of those trophozoites inside of the red blood cells. You do blood smears, and you look for the presence of the trophozoites inside of the red cells. This is an intracellular parasite. So you're looking for the trophozoite literally inside of the red blood cells. And you're looking for patient symptoms and signs such as recurring chills, fever, and sweating. Treatment includes these quinine drugs, chloroquine, and artemisinin-based combination therapy. Control includes mosquito decline and human prophylaxis, mosquito nets even. There's even a vaccine that's now available as well. Here's an image of those sleeping nets I told you about. Insecticide-treated bed nets, right? These keep the mosquitoes off during sleep. Now, moving on with these alveolate parasites, cause zoonotic disease in domestic animals and birds, which can cause severe diseases in humans. These can exist in several stages, including dormant oocytes and pseudocytes. Let me give you an example. Toxoplasma gondii, which causes toxoplasmosis. This is a really interesting one, especially since I love my cats so much. This one's linked to cats, people. So if you're a cat person, you may want to pay attention to this one. So this is an intracellular apicomplexan parasite. It lives naturally in cats that harbor oocytes in their GI tract. These oocytes are eggs. People acquire this Toxoplasma gondii parasite by ingesting raw meats or substances contaminated by cat feces. Most cases of toxoplasmosis go unnoticed, except in the fetus and in AIDS patients who can suffer brain and heart damage. These are two treatments that you can take for the disease. This is also why, if you happen to be pregnant, they tell expectant mothers not to scoop the cat box, right? Because if your cat were to have this toxoplasma, it could affect their fetus. So in this image here, you can see the life cycle of Toxoplasma gondii. You can see that, let's say a mouse here, this mouse possesses the pseudocysts of this protozoa. If your cat were to hunt down this mouse and eat it, it would pick up those cysts, and those pseudocysts would now be present inside of the cat. And when the cat uses their litter box, they can pass this protozoa into their feces, and that's where sporogeny occurs. Sporogeny is where the pseudocysts form into oocytes. They form the eggs. And if a person were to ingest those eggs, that's how it would infect the person and affect their brain if have potential damage to the brains of the fetus or to compromised AIDS patients. Next, concept check number 5. A person can acquire toxoplasmosis from what? Pseudocysts in raw meat, yes. Oocysts in the air. Well, I guess if you're cleaning the cat box and it kicks up the oocysts, yes. Cleaning out the cat litter box. So I would say all of these. All of these. And that's why it's so important not to eat raw meat or clean the kitty litter box if you are an expectant mother. All right, now, before we move on to helminths, remember helminths are worms. We're about at the halfway point, and we need to do a break time, right? We need break time with Gizmo and Wicket, see what they're up to. Come back strong with helminths and tell you about some worm infections that are even more gross somehow than the protozoal infections. Right, Punky? Yeah, and Punky's gonna help. All right, welcome back from break time with Gizmo and Wicket. Let's talk about helminths now. Helminths refer to worms, and adult worms are large multicellular animals with actual specialized tissues and organs. Adult worms can mate and produce fertilized eggs that hatch. Larvae then mature in several stages to adulthood. The sexes may separate or be hermaphroditic. So some worms have a clear male and female sex. Some worms possess the sperm and the eggs in the same worm. That's what they mean by hermaphroditic. Adulthood and mating occur in the definitive host. The definitive host is the animal or the host that, where adulthood and mating take place. However, larval development occurs usually in an intermediate host. A transport host experiences no parasitic development. And we're going to cover these. There are 4 basic patterns of life and transmission for helminths. And do you remember from a previous chapter, we talked about how there are the roundworms, also known as nematodes. And then there are the flatworms, remember the platyhelminthes, which include the trematodes, also known as flukes, and the cestodes, which are known as tapeworms. And remember, there are 4 life cycles. Let's talk about cycle A. Here, the worm is ingested through the food or water. The larvae then hatch and enter tissues. The worm develops in the intestine. Egg is released with the feces into the environment. The eggs are then ingested by a new host. Examples of this life cycle are exhibited by worms Ascariasis and Trichomonads. Here's another life cycle, cycle B. In this case, infection occurs through skin penetration by the larvae. So you see, for instance, hookworms can enter the skin through the foot, for instance. Those larvae then enter tissues and migrate. They migrate to the intestines where they are passed in the feces. And it's those eggs that are released with the feces. The eggs hatch into larvae. Those larvae develop in the environment and then again get through the skin with penetration through the skin. All right, here's cycle C. A human eats contaminated meat that contains cysts from a worm cysts. And an example here is Tania, or the tapeworm. So this poor person is about to eat tainted meat that's full of tapeworm cysts. Those tapeworm cysts are released by the stomach acids. The stomach acids cause the cyst to open up. The larvae then enter the intestines, and those tapeworms attach to the intestinal lining. Those tapeworms produce fertilized eggs. The eggs are released into the environment and are eaten by grazing animals such as cows. In this case, the human is the definitive host where adulthood and mating occurred, and the cow is the intermediate host where larval growth occurs and encystment occurs. So this means that the larvae form into cysts within the muscle of the cow. And then if the person eats that tainted meat, they will pick up more cysts, right? They're going to ingest more tapeworms. And the 4th life cycle, cycle D, in this case, the human, the definitive host, is infected either through ingestion or through direct penetration of the skin by the larval phase. Examples of these worms are Apis and Schistosoma. Those worms migrate to the intestines or the bladder. Eggs are formed. Those eggs released larvae. Those larvae are secreted in the definitive human host. There's the fertilization to form eggs. Those eggs are secreted usually through the feces. Those eggs hatch into the first larval stage, which occurs in the environment. And then the second larval stage usually involves some sort of intermediate hosts. And again, by either consuming animal flesh of an intermediate host or through direct skin penetration, that's when the new human, the new definitive host, picks up the larvae and it becomes an adult. So I hope you're noticing that although worms are quite big, it's usually the tiny eggs or the larvae or the cysts that are quite small that are what infects our bodies, either through usually ingestion or through penetration of the skin. And once those worms are inside our bodies, pathology, the disease, arises from those worms feeding on and migrating through our tissues. The accumulation of worms and worm products can cause damage to our tissues. To diagnose a worm disease, we can either base it on a blood cell count, eosinophilia. Remember, the eosinophils are the white blood cells that attack helminths, and so they should be higher or elevated if you have a parasitic infection. Serological tests, remember, these are clinical tests based on antibodies binding to antigens. Looking at the eggs, did you know there are entire fields of microbiology dedicated to understanding what these eggs look like and how the eggs work? So a true microbiologist could look at a worm egg and determine which species it might be simply from the shape of the egg. the larvae or the adult worms in the feces, sputum, urine, blood, or tissue biopsies. Treatment include drugs that are called anti-helminthic drugs. These can suppress a helminthic metabolic process that differs from the human process, targeting those worm-specific metabolic processes. It can inhibit the worm's movement, prevent it from holding position, or act locally in the intestine where the worms are. Here's a table of anti-helminthic therapeutic drugs or chemotherapies. And you can see, obviously, you don't need to memorize all these, but it's nice to see, the variety of different drugs and kind of what they interfere with and what worms are affected. So again, depending on what you're diagnosed with, what type of helminthic infection, you're going to be prescribed one of these various drugs. So again, remember, nematodes are the roundworms. These are the most abundant animal groups. The vast majority of these are free living in the soil, freshwater, but around 200 of these are parasitic, and 50 of those affect humans. And do you know what roundworm means? It means you have a round worm, an elongated cylindrical worm with protective cuticles, circular muscles, a complete digestive tract, and they have distinct separate sexes. So you can tell the difference between a male worm and a female worm. The parasites are divided into two groups: intestinal nematodes and tissue nematodes. The intestinal nematodes develop to some degree in the intestine. This includes Ascariasis, strongyloids, and hookworms. Tissue nematodes spend their larval and adult phases in various soft tissues other than the intestines. For example, various filarial worms. So here we can see some examples. Intestinal nematodes that have cycle A type life cycles. Remember cycle A? Cycle A, where you ingest the eggs hatch, forming larvae inside of the intestines, and then you poop, the feces release those eggs, and then, the egg matures and gets, and that's the cycle A. This includes worms like Ascaris, lumbricoides, Trichuris, Trichuria, and Enterobius vermicularis. There are cycle B worms. Remember cycle B? In cycle B, the egg is passed in the feces, the egg hatches into an early larva, and then that infective larva penetrates through the skin. Remember that? And then those larvae migrate to various tissues and ultimately get into the intestines where they're passed again. This includes hookworms, strongyloides, and Trichinella spiralis, which causes Trichinellosis. And then remember, there are the tissue nematodes. These are the filarial nematodes. For example, Wuchereria bancrofti, which causes bancroftian filariasis. Let's talk about Ascaris lumbricoides, and it causes Ascariasis, a large intestinal roundworm. Most cases in the U.S. occur in the southeastern states. It's indigenous to humans. Ascaris spends its larval and adult stages in humans. They release embryonic eggs in the feces and are spread to other humans through food, drink, and contaminated objects, right? So fecal contamination. Ingested eggs hatch into larvae and burrow through the intestine into your circulation. They could travel to the lungs, the pharynx, and are swallowed. Adult worms complete cycle in intestines and reproduce 200,000 eggs per day. And again, you would then release those in the feces. Those eggs are released in the feces. These worms are interesting. Unlike tapeworms, for instance, they do not attach. They retain their motility. They cause severe inflammatory reactions, marking the migratory route they're taking through your body. Allergic reactions can occur, and heavy worm loads. Remember, this is a large worm. Heavy worm loads can retard physical and mental development. Here you can see just how large these worms are. They are as long as this person's hand. However, remember, it's those eggs that are ingested and passed on in the feces, and these eggs are microscopic. Here's a microscopic view of a fertile Ascariasis egg, and here's the larva on the right from the fecal sample. Next, another roundworm with cycle A. This is Enterobius vermicularis and pinworm infection. Pinworm or seatworm is what it's called. Enterobiasis is most common worm disease in children in temperate zones. You typically see pinworm infection in children. Eggs are picked up from surroundings and swallowed. After hatching in the small intestine, they develop into adults, and that could lead to anal itching when mature females emerge from the intestines to release eggs. And then the way that this progresses, this infection progresses, is that if you use the bathroom and don't wash your hands, or if you are not hygienic, like most little children are not, they will get fecal matter on their hands, and then they can self-inoculate themselves again, which means perpetuate the cycle of reintroducing the worms into their system, and they could even spread it to other children as well. So let's say they have fecal contamination on their hands, they've got these eggs on their hands, they can then touch other objects with which other children touch, and so daycares are usually big incubators for spread of pinworm disease. And concept check number 7, humans become infected by which stage of the pinworm's life cycle? Remember, it was those eggs. Remember here it says anal itching occurs when mature females emerge from the intestine, from the anus, in order to release eggs. Next, let's talk about another nematode, but remember, this is a cycle B type. Hookworms have characteristic curved ends and hooked mouths. Necator americanus and Ancylostoma duodenale are examples of hookworms. Humans shed the eggs in the feces, which hatch into fowleriform larvae. These burrow into the skin of bare feet. Remember, cycle B type worms, they can penetrate skin. So here's some depictions of hookworms. The male hookworm is shorter than the female hookworm. Here's a close-up of those hooks in the mouth. You can see the cutting teeth. Here, this is N. americanus, and at the bottom you have A. brasiliense. Notice how these are hooked mouths. And then you also have the egg stages in the feces. So once those larvae penetrate through your skin, they can travel from the blood to the lungs, proceed up the bronchi and throat, and are then swallowed from the throat. Worms mature and reproduce in the small intestines and complete the cycle. This may cause pneumonia, nausea, vomiting, cramps, bloody diarrhea. Blood loss is significant as well. This is known as anemia. All right, here's another type B cell life cycle worm that's a nematode called Trichinella spiralis, which causes Trichinellosis. A person picks up Trichinella by eating undercooked meat, usually undercooked pork, but it could also be other infected animals as well. When you do ingest that meat, usually you are ingesting the cysts of Trichinella. Larvae are then released in the small intestine. Adults form in the small intestine, and then larvae are deposited in the mucosa, and you could end up with encysted larvae in your striated muscles. Unfortunately, you could get these worm cysts in your muscles. Again, these worms are acquired by eating undercooked pork or bear meat. You're eating those encysted larvae. The larvae migrate from the intestine to blood vessels. They can migrate to the muscle, the heart, the brain, where it then forms cysts, larval cysts. The first symptoms of this infection are flu-like symptoms, diarrhea. Secondary symptoms include muscle and joint pain, shortness of breath, and pronounced eosinophilia. Remember the numbers of eosinophils in the blood. If you have a worm infection, you're going to have more of these white blood cells in your blood. There is no cure after the larvae have encysted. Oof, so you don't want to be eating undercooked pork or bear meat. All right, next let's talk about the tissue nematodes. Remember, these are the filarial worms that cause filariasis, a chronic deforming disease. Let me show you an example. Wuchereria bancrofti, which causes elephantiasis. This is a tropical infection spread by mosquitoes. Mosquitoes are the vector that deposit larvae, which move into your lymphatics and develop in adults. Mosquitoes are the vector that deposit worm larvae on the host, on the human. Those worms migrate to the lymphatics and develop into adults. Chronic infection causes blockage of the lymphatic circulation. Remember how your lymph vessels have to circulate? They have to, whenever you do muscle contractions, the lymph fluid is supposed to migrate towards your heart. Well, guess what? These worms get in the way and block lymphatic circulation, and that results in fluid buildup in your lymph vessels, and this results in a condition known as elephantiasis, massive swelling in the extremities. You see this on the left leg of this individual. This is a sign of that disease, elephantiasis, and it's due to Wuchereria bancrofti getting in there and blocking the lymph ducts. Here you can see what the worm looks like from a blood smear. Here's another interesting worm I wanted to tell you about. It's a roundworm. This is Dracunculus medinensis, and it causes draconiasis. Draconiasis is caused by that worm I just told you about. It's also known as the Guinea worm. It affects residents of India, Middle East, Central Africa. This worm is a parasite carried by Cyclops, a microscopic arthropod commonly found in still water. If a person were to ingest Cyclops, they would also ingest this guinea worm. After ingesting water from a contaminated water source, the larvae of the worm, the guinea worm, are freed from their Cyclops host and penetrate into subcutaneous tissues of the intestine, where they form highly irritating lesions. And what's worse, those worms can then burrow through, they can leave the intestines, burrow through your body, through your muscles, down to your lower extremities. I'm talking about your legs. And if you visit a watering hole like a pond or a stream, the worm will protrude from your skin. It will burrow out of your skin where it deposits its eggs into that stream. Isn't that gross? And then the only way to get the worm out of your body is to wrap it around a stick very, very slowly, a tiny bit each day, until you have extracted the entire long worm. Next, let's move on to the flatworms. Do you remember that the flatworms are called Platyhelminthes? And they include the trematodes and the cestodes. So let's talk about the trematodes first, one of the types of flatworms. These are flatworms with ovoid, leaf-like bodies, and some of them really do look like little leaves. I'll show you. They have a digestive, excretory, neuromuscular, and reproductive system. They lack a circulatory and respiratory system. And with these trematodes, animals such as snails or fish are usual suspects as intermediate hosts, and the humans are the definitive hosts. So many people pick up trematode infections by eating snails or fish that are not cooked. Also, another word for trematodes, remember, were called the Flukes. Trematodes are flukes. And one example of these flukes are called schistosomas. These schistosomas cause diseases called schistomiasis. Examples are Schistosoma mansoni, Schistosoma japonicum, Schistosoma hematobium, and these are cycle D worms. Remember, let's go back. Cycle D worms can either be ingested or penetrate through the skin. With schistosoma, remember that the human is the definitive host, so the adult fluke lives in the human. The human then passes those fluke eggs into a water source. That forms an early larval stage called Miracidium, and that forms inside of a snail. A snail picks up the Miracidium, where inside of the intermediate host, this snail, the worm forms into a second larval stage, the Cercaria. Those larvae can then penetrate human skin and mature into the liver. The adult worms migrate into the intestine or bladder and shed eggs. This can cause chronic organ enlargement. Here's what schistosomes look like. This is the Miracidium phase, which infects the snail. So this is what the worm would look like if cultured from a snail. Here you can see the Cercaria phase, which is released by the snails, and that can burrow into the human host. And here's what the adult schistosoma looks like. There is a male and a female gender. The long, slender female worm is cradled within a groove on the ventral canal of the larger, thicker male worm for mating. So this is the characteristic mating of the schistosoma worms. Remember, the long, slender worm is the female, and the wider one is the male. There are also trematodes or flukes of zoonotic origin. This means they come from animals. Apis thorchis sinensis is an example. This one cycles between mammals and snails and fish. Humans are infected by eating inadequately cooked fish containing Cercariae. Those larvae crawl into your bile ducts, mature, and shed eggs into your feces. Snails are then infected, and the life cycle can repeat. Fasciola hepatica. This is the liver fluke. It cycles between herbivores, such as cows, snails, and aquatic plants. Humans are infected by eating raw aquatic plants. The fluke lodges in the liver. That's how it got its name, the liver fluke. Here's what the liver fluke looks like. It really does look leaf-like, doesn't it? Remember, some trematodes look leaf-like in appearance? And this fasciola is interesting. Unlike schistosoma, which remember had the long, slender female and the wider grooved male, fasciola, which is another type of trematode, is hermaphroditic, which means it has both sex organs within the same worm. It can make the sperm and the eggs within the same worm. And the lung fluke, Paragonimus westermanni. This lung fluke cycles between carnivorous animals, snails, and crustaceans to humans. Humans get infected by eating undercooked crustaceans. So another important reason to cook your food. It's a great way of picking up parasitic infections if you eat raw or undercooked foods. These worms then leave the intestinal lining and migrate to the lungs. And concept check number 9. The organism that causes elephantiasis has an enclosed digestive tract. That is true. And then the last type of worm, remember, it's another type of flatworm or platyhelminthes called cestodes. Cestodes is another term for tapeworms, and everyone has heard of tapeworm infections. These worms are long. They're very thin, ribbon-like. That's how they got their name, tapeworms. They look like tape, like measuring tape or something. They've got ribbon-like bodies composed of sacs or segments called proglottids and a Scolex head. The head is called the scolex, and the body segments are called proglottids. The scolex grips the intestine. That's how the tapeworm sticks to your intestines. Each proglottid is an independent unit adapted to absorbing food and making and releasing eggs. This is another type of hermaphroditic organism. Tapeworms can produce both the sperm and the eggs, so you only need to be infected by one worm in order to make fertilized reproduction and make eggs, fertilized eggs. These worms are the principal cause of human Taeniasis. These are the two main genuses. These are the two main species that infect humans. Taenia saginata, the beef tapeworm, and Taenia solium, the pork tapeworm. Taenia saginata is interesting. It can become so big. It's the beef tapeworm. It can become very large, meters long, up to 2,000 proglottids. Humans are the definitive host. It's where the worm becomes an adult and does sexual reproduction with itself, right? Because it's a hermaphroditic organism. By the way, this right here is the scolex of the pork tapeworm, Taenia solium. It's characterized by 4 suckers. Yeah, 1, 2, 3, and then the fourth one's on the backside, and a rositellum containing 2 rows of hooklets. See, 2 rows of hooklets for the pork tapeworm. The beef tapeworm, Taenia saginata, on the other hand, lacks these rostellar hooks and attaches to the intestine using suckers alone. Again, Taenia saginata, this is the beef tapeworm. Animals get infected when they're grazing. So let's say the cattle graze on the plant matter. If that plant matter is contaminated by human feces, remember the eggs are released by the human in the feces. People are then infected by eating raw beef because that beef contains the cysts, the larval cysts. And if you ingest the larval cyst travels to your stomach where the cyst breaks open. In humans, the larva then attaches to the small intestine to become an adult. And remember, once it becomes an adult, it can form fertilized eggs because it's hermaphroditic. It causes few symptoms. Some people can have tapeworms, or especially the beef tapeworm, and never know. There's vague abdominal pain and nausea. You can even have proglottids in the stool, like parts of the tapeworm could break off and appear in the stool. Here's generalized diagram of the life cycle of beef tapeworm. Again, you ingest the encysted larvae. Those cysts break open in the stomach acids. The stomach acids help release the larvae. The larvae then stick to the intestinal lining with their scolex head, either with their hooks or the suckers, or both. And then it forms into the mature adult worm. These are proglottids, right? These are proglottids, which can form the sperm and the eggs. And then those eggs are released in the stool. If those eggs contaminate the food supply of bovine or cattle, those cattle will ingest those eggs. Those eggs form cysts inside of the muscles of the cattle. And then again, if you eat undercooked meat, the cycle continues. Next, let me tell you about the slightly more dangerous pork tapeworms. If you're going to have a tapeworm, you want to have beef tapeworms. Believe me, you don't want to have pork tapeworms, and I'm going to explain why. Pork tapeworm, or Taenia solium, infects humans when they ingest those cysts. Remember the encysted larvae or eggs. This is again from undercooked pork. In cysticercosis, eggs hatch in the intestine, releasing tapeworm larvae that migrate to all tissues and encyst. Okay, this is not good because unlike the beef tapeworm, when the pork tapeworm is re-ingested. Let's say you ingest pork tapeworms. The first tapeworm will probably reside in your guts and it'll become an adult, right? That adult tapeworm will release eggs. And if you were to not wash your hands and re-inoculate yourself with those eggs could then migrate to all tissues and form cysts, a process known as cysticercosis. You don't want this because those cysts can end up everywhere in your body. Imagine worm cysts throughout your body, like riddling your body. The most damaging worm cysts occur if they lodge in the heart, the eye, or the brain. And yes, this can often happen with people who enjoy eating undercooked bacon, or if they eat undercooked pork. It's not uncommon to have these cysts show up in your brain. And that is the main cause of adult onset seizures. Did you know that? This may cause seizures, psychiatric disturbances. Here's some pork, and you can see all those cysts inside of the porcine muscle. All right, and on that bombshell fun stuff, let's move on to arthropod vectors. Arthropods have exoskeletons and jointed legs, including insects, arachnids, and crustaceans. Many must feed on blood and tissue fluid of host during life cycle. These are known as ectoparasites. So, again, arthropods, I think of them as biting insects usually. These are great vectors. Vectors are living transmitters of disease. Think a mosquito. Think a tick, right? Things like this are arthropods. And many of them transmit infectious microbes in the process of feeding. So they are biological vectors. Do you know the difference between insects and arachnids? Insects have 3 body segments and 6 legs, while arachnids have 2 body segments and 8 legs. So let's just touch on some of these. Mosquitoes. Everyone has seen a mosquito. Mosquitoes require an aquatic habitat because when they lay their larvae, they do so in water. This is why it's so important to get rid of standing water near where you live. They lay their larvae in water, standing water. And it's usually the female mosquito that spreads the disease. Females are the ones that take a blood meal. They're the ones that feed on blood and transmit disease, including malaria, filariasis, dengue fever, Zika virus. Okay, and fleas. We've all heard of fleas. Usually it's a dog or a cat that catches fleas, but fleas are highly motile with flattened bodies. They feed on warm-blooded animals. They can carry zoonotic diseases. Remember, the plague was spread by fleas. Marine typhus is spread by fleas. Marine means mouse. Lice. Lice in the hair. You've heard of people with lice. Small, soft, attached to head and body hair, feeding inconspicuously on blood and tissue fluid. Well, they can release feces and contaminate wounds. They can cause epidemic typhus and relapsing fever. Here you can see a mosquito feeding on a human. And how many diseases are spread by mosquitoes? It's countless diseases are spread by mosquitoes. A flea, this is what a flea looks like on the right. Too bad he's covering his little mustache. A lot of fleas have this characteristic little mustache looking thing. It's part of their mouth structure. Here you can see a louse. A louse is singular for lice. So these are all insects. But remember, arachnids have 8 legs. And ticks are a big example of arthropods that are arachnids. Ticks, these guys cling on vegetation and attach to a host on contact. The larvae, nymph, and adults get blood meals by piercing skin of host. So these little guys, they get onto your body and then they feed on your blood. There are hard ticks and soft ticks. Hard ticks are known as ixodid ticks. Soft ticks are known as argacid ticks. The hard ticks are small, compact, with rigid bodies. They transmit bacterial, rickettsial, and viral diseases. The soft ticks got their name because of their flexible outer bodies, and they can transmit relapsing fever. And here you can see a hard tick. In this case, it's amblyoma. Here you can see A, B, and C are the feeding stages, and D and E are the stages of engorgement, right? So these are the ticks themselves. This is what that tick looks like after it's fed. It can grow several times its size because it's engorged itself with blood. Here's a soft tick. You can see the ventral and the dorsal view. Ventral means the bottom side of the tick, and dorsal means the top side of the tick. And that's it. We did it. We got through a lot of really, really interesting eukaryotic parasites, as well as ectoparasites, those arthropods that bite. A pretty long chapter, but a very interesting chapter. I hardly wanted to cut anything out because it's so interesting to learn about all these diseases and how they're spread and what parasitology is all about. Again, if you ever take a parasitology course, you'll learn so much more about all of this from this chapter. What an interesting chapter. I believe the last two chapters we're going to cover are going to cover viruses. So let's go ahead and get over to that chapter.
Dr. D, Dr. D, Dr. D. 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 Dr. D, Dr. D, Dr.